Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. For decades, we thought we had the human genome mapped, right? A complete blueprint. But what if I told you we were essentially looking at a pixelated image, missing massive, crucial chunks?
Yeah.
Today, we're diving into a groundbreaking study that completely shatters that illusion. Imagine discovering not just a few new genes, but over 200 million new letters of human DNA sequence,
It's incredible.
finding tens of thousands of structural variations previously invisible, and even finding chromosomes that might have two key attachment points. It's like finding entire lost cities within our own genetic code. Now, you might recall the Human Genome Project. Monumental achievement, gave us our first human blueprint.
Absolutely. A huge step forward.
It was staggering for its time, but here's the kicker. Huge repetitive regions, like the ends of chromosomes, the telomeres, and the centromeres, they were just left as gaps.
Yeah, they were basically unmappable back then.
Think of them as blank spots on the map. We were missing large chunks of structural diversity. We had the highways, sort of, but none of the complex neighborhoods.
Exactly. And these missing regions weren't just empty spaces. They were incredibly complex, really challenging to sequence with the older technologies.
Right.
So this meant we couldn't fully understand things like structural variants, large-scale changes in our DNA like deletions, duplications, inversions, where whole segments get rearranged.
Okay.
Now, newer long-read sequencing technologies, they started to bridge some gaps, sure, like PacBio Hi-Fi and Oxford Nanopore ultra-long reads. But getting a truly complete telomere-to-telomere picture, a full unbroken sequence from end to end, especially for a diverse set of human populations, that remained a major challenge.
And that's where this study comes in.
That's exactly why we needed this deep dive.
Okay, so our mission today is to unpack this groundbreaking Nature article. It leveraged really cutting-edge technology to assemble a collection of nearly complete human genomes.
That's the goal.
We'll explore what they found in these previously dark regions, how it changes our understanding of human biology, disease, and what exciting new research paths it opens up for all of us. Building the blueprint, assembling nearly complete genomes. So how did they finally crack these notoriously difficult regions? It sounds like a massive undertaking.
It absolutely was. The first step was getting a really broad spectrum of human genetic diversity.
Okay.
They meticulously selected 65 diverse individuals representing 5 continental groups, 28 distinct population groups.
Wow, that's broad.
Yeah. And then they combined the strengths of different long-read sequencing technologies. Think of it like using different kinds of magnifying glasses, right? A PacBio Hi-Fi reads super accurate and ultra-long Oxford Nanopore, or ONT reads, which can span huge distances.
Got it.
And they didn't stop there. They integrated other powerful methods too, like StrandSec, Hi-C, optical mapping, all crucial for piecing together these complex puzzles that older methods just couldn't solve.
So, after all this, did they really manage to fill in those massive blank spaces? What did the results actually show? Did they close the gaps?
The results were, well, truly remarkable. They generated 130 highly continuous haplotype resolved assemblies.
Okay, haplotype resolved. That's important, right?
Yeah.
Separating mom's and dad's DNA.
Exactly. It means they could actually tell which DNA came from which parent. So you get 2 distinct, incredibly accurate copies for each person, not just some blended average.
Nice.
And the contiguity, like how long the unbroken stretches of DNA were, astonishingly high. A median of 137 megabases, that's basically continuous sequence for huge chunks.
Wow.
And the accuracy, the base pair accuracy, incredibly high. Like the genetic letters they read were almost perfectly correct.
That's amazing. So they effectively closed 92% of all previously reported gaps.
92%, yeah.
And for nearly 40% of chromosomes, they got telomere-to-telomere status.
Wow.
602 chromosomes assembled as single gapeless sequences.
602, it's incredible.
Imagine finally filling in those blank spots on our genetic map with real detail instead of just, guessing what was there.
So yeah, this immediately raised a big question. What about all the genetic variations we thought we knew?
Right, the ones we thought we'd already mapped.
Exactly. When they compared their new complete T2T CHM13 reference genome to the old one, GRCH38, they identified a staggering 188,500 structural variants, those big rearrangements,
Whoa.
plus millions of indel small insertions or deletions and single nucleotide variants, SNVs.
Okay.
But think about that number for the structural variants. It represents a 59% increase in detection compared to the older methods.
59%. That's huge.
It is, and at the same time, a 55% reduction in false discoveries.
So much more accurate and finding way more.
Precisely. What it means, basically, is that we were missing well over half of the large structural variations that fundamentally shape who we are.
That's not just a small correction.
No, it's a profound shift in our understanding of human genetic diversity and what causes variation between individuals. 2. Uncovering hidden genomic diversity beyond the basics.
Okay, that is a huge revelation. So with these new, more complete maps, what specific hidden gems did they find? Stuff we just couldn't see before. Let's start with those jumping genes, mobile elements.
Indeed, mobile element insertions, or MEIs. These are segments of DNA that can copy and paste themselves into new spots.
Right.
They make up a significant chunk of this newly discovered variation. They found nearly 13,000 new MEIs. That's a 36% increase.
Wow.
And a lot of that increase came because they included more individuals of African descent whose genomes generally have more diversity.
That makes sense.
And what's fascinating here is that the vast majority of these full-length L1 insertions, they still seem to retain their ability to retrotranspose.
Meaning they can still jump.
They can still jump to new locations in the genome, yeah.
What?
Potentially creating new variations even now.
Okay, what about inversions? Where a piece of DNA flips around, that can really mess things up, right?
It certainly can. They identified 276 inversions, and 21 of those were completely novel, never seen before.
Okay. Any examples that stood out?
Yeah, one notable one was a large 1.8 megabase inversion on chromosome 5. It's in a region known to overlap with the Sotos syndrome critical region.
So potentially disease relevant.
Exactly. It shows how crucial resolving these complex structural changes can be for understanding genetic disorders, maybe finding your origins, leading to better diagnostics, stuff like that.
What about segmental duplications?
It is.
These are those large, almost identical chunks of DNA copied multiple times. Super tricky to map accurately, I imagine.
Incredibly tricky. They found an average of about 168 megabases of SDs per genome, and these new assemblies gave a much, much better picture of interchromosomal SDs.
The ones that jump between chromosomes.
Yeah, adding another layer of complexity. But critically, they identified 23.6 megabases of novel SD sequences we just didn't know were there.
23 million new base pairs of duplications.
That's right. And a significant chunk, 2.4 millibac, was on the X chromosome, overlapping 167 protein-coding genes.
So this isn't just like junk DNA, it overlaps actual genes.
Absolutely. It's like discovering new genetic material within our own blueprints.
And did these novel duplications differ across populations? Does it reinforce why we need diverse samples?
Yes, absolutely. They found that genomes with African ancestry, on average, had more of this novel SD content compared to non-African individuals.
Okay.
It just hammers home how critical it is to study diverse populations. If you don't, you miss a huge part of the picture of human genetic variation. Your reference genome ends up biased.
Now, the Y chromosome, always notoriously difficult, right? So repetitive.
Yeah.
Did these new methods finally help unravel its secrets?
They really did. They got highly contiguous Y assemblies for 30 male individuals, and for 7 of them, the assembly was unbroken across the entire male-specific region.
Wow, seamless Y chromosomes.
Pretty much. And this let them finally investigate the biggest heterochromatic region in the genome, YQ12. It's made of these highly similar but size variable alternating repeat arrays.
A region that was largely a mystery before.
A total mystery, yeah. A real genomic dark spot.
So what does this tell us about the Y? What did they learn about its variability between men?
Well, they found huge variation in the number and length of these repeat arrays in YQ12, ranging from about 18 megabases up to over 37 megabases.
That's a massive difference in size.
Huge. And they could even use the unique patterns of tiny ALU insertions, another type of mobile element, kind of like molecular breadcrumbs, to track the dynamic evolution of this region over time. Gives us vital clues about its history and how it changes.
Okay, this is amazing mapping, but it's not just about the map, right? It's about function. What do these structural variants do? How do they affect our genes, our biology?
That's the crucial question. If we connect this to the bigger picture, they found that 1,535 of these structural variants directly disrupt 985 unique protein coding genes.
Disrupt them how?
Well, imagine a gene is like a recipe, right? An SV isn't just changing a word. It can tear out a page, duplicate a paragraph, completely change how the final protein is made.
Okay.
For instance, they found a common deletion in a gene called ZNF718. This one deletion fundamentally rewrites its instructions, leading to 9 unique versions or isoforms of that protein.
9 different proteins from one gene, just because of one deletion.
Exactly. It's a powerful example of how one previously hidden structural variant can drastically alter gene products and potentially function, leading to all sorts of biological differences.
What else did they find linking these SVs to function, to actual biological effects?
So they found 122 unique SVs located near 98 differentially expressed genes.
Meaning genes that are turned up or down.
Right. These SVs are associated with changes in gene activity levels, and some even corresponded to changes in chromatin conformation.
How the DNA is packaged.
Exactly. How it's folded up in the nucleus, which directly affects whether genes can even be read.
Okay, so this raises a big question.
It does. How many common diseases could be linked to these structural variants that we were just missing before?
Right.
And in fact, they directly tackled that. They identified 3,818 SVs that are in high linkage disequilibrium, basically, closely statistically linked with SNPs from previous GWS studies of human disease.
So connecting the dots between these new variants and known disease associations.
Precisely. It's filling in a major piece of the puzzle for lots of human traits and conditions. 3. The new era of genotyping, pangenomes and precision.
Wow. This whole project feels like it's laying the groundwork for a completely new way of looking at our genomes, moving beyond just one reference map. What does that future look like?
Absolutely. This study is a huge contribution to building what we call a pangenome reference.
Not just one genome, but a collection representing diversity.
Exactly. The full tapestry of human diversity. They integrated their 65 new assemblies with 42 others to create this pangenome graph, and then use that graph to genotype over 3,200 individuals from the 1,000 Genomes Project using standard existing short-read data.
Okay, using the new map to better interpret old data.
Precisely.
And what's the tangible impact of that? For finding variants, especially the rare ones.
For you, the listener, this means a huge leap in precision. Using this new pangenome approach, they detected an average of 26,115 structural variants per genome.
That's way higher than before.
Significantly higher. And crucially, this dramatically increased the detection of rare structural variants, especially in individuals of African descent.
How much of an increase?
They found, on average, 1,490 rare SVs per genome in individuals of African descent, compared to only 477 previously.
That's more than triple.
Yeah, it directly addresses those historical biases in genomic studies, making genetic research far more equitable and, frankly, more accurate for everyone.
And did this help reconstruct entire personal genomes more accurately, even from that older short-read data?
Yes, significantly. They could reconstruct highly accurate consensus haplotypes, those complete mom and dad DNA copies, achieving really high fidelity from short reads. Big improvement. And for targeted genotyping of complex, medically relevant genes, like the HLA system, crucial for immunity and transplants.
Yeah, that's super complex.
Right. This expanded reference panel boosted the accuracy there to over 86%.
That's a game changer for things like transplant medicine or understanding autoimmune diseases.
Absolutely. Where precise genetic understanding can be life-saving. 4V, deep dives into genomic hotspots, MHC, complex SVs, and centromeres.
Okay, let's zoom into some of these genomic hotspots.
Yes.
The major histocompatibility complex, MHC, super complex, vital for our immune system. What did these complete assemblies show there?
Yeah, the MHC is a beast. It's a 5 megabase region just packed with genes, incredibly difficult to resolve fully.
But they managed.
They did. Using these new assemblies, they could properly annotate between 27 and 33 HLA genes. Those are the key immune genes, and they resolved 826 incomplete HLA allele annotations that were already in databases, basically cleaning up and completing the picture. They also found 170 novel structural variants just within the MHC, including a whole deletion of one HLA gene, HLA-DPA. Shows the fine-grained detail they achieved.
That's incredible mapping. But how did having these complete sequences help understand the evolution of these HLA types? They're so important for immunity.
Right, it's like a puzzle box, the MHC. But with the full sequences, we finally got a clearer picture of how different HLA types evolved.
Like how?
Well, for example, they showed how patterns of repeat elements suggest certain HLA types probably arose from specific deletions or recombination events way back when.
Seeing the history in the sequence.
Exactly. And they tackled this notoriously complex cluster called RCCX, revealing common duplications and rare, really intricate gene conversion events that shape its evolution.
So getting at the mechanisms driving diversity in this key immune region.
Precisely. It sheds crucial light on how these vital regions evolve and contribute to our individual immune profiles and, susceptibility to different diseases.
Now, beyond simple changes, they also looked into complex structural variants, CSVs. What exactly are those, and what did they find that was hidden before?
Okay, so CSVs are basically single genomic events that involve multiple simple structural variants all tied together.
Like a deletion, an inversion, and a duplication all happening at once.
Kind of, yeah. All interconnected in one complex rearrangement. And their breakpoints, where they start and end, are often in highly repetitive sequences, making them incredibly hard to spot.
Okay.
They found an average of 72 CSVs per genome, over 1,200 unique ones across the whole dataset. Many involved local duplications or inversions bundled together.
Can you give us a concrete example, something that shows why resolving these CSVs actually matters?
Sure. One fascinating example involves the NOTC H2NL and NBPF genes. These have been implicated in human brain expansion.
Oh, interesting.
Yeah. They were able to resolve 3 distinct haplotypes in this region, including a really complex 930 kilobase CSV involving an inversion and deletions. Previous methods just couldn't piece it together.
Okay, and another one, maybe disease-related?
Absolutely. The SMN1 and SMN2 gene copies linked to spinal muscular atrophy, SMA.
Right, that's a well-known one.
It is, but despite decades of research, fully sequencing this region was almost impossible because of its complexity. But this study, they successfully assembled and characterized 101 distinct haplotypes, finally resolved the complex structure and copy number variations.
So what does that mean for understanding SMA risk? Or even human evolution in the other cases?
Well, for SMA, they found that 98 of these haplotypes carried the healthy SMN1 copy, but 3 did not.
Potential disease risk haplotypes.
Exactly, likely arising from gene conversion events. This has direct implications for understanding SMA risk and potentially for treatment.
Okay.
And then they looked at the amylase locus. That impacts starch digestion. It's highly variable depending on ancestral diets.
Right, people have different numbers of amylase genes.
Exactly. They identified 39 distinct amylase haplotypes, capturing about 83% of the population's variation, and they fully resolved the largest known haplotype, which has 11 copies of the AMY1 gene.
Wow. Connecting genetics directly to dietary adaptation.
Precisely.
Okay, now for maybe one of the most surprising findings, centromeres. The crucial centers of our chromosomes, essential for cell division, but notoriously hard to map. What did they uncover there?
Yeah, centromeres are among the most changeable, mutable regions in the genome, just walls of highly repetitive DNA.
It's a huge challenge.
The huge challenge. But this study completely and accurately assembled an astonishing 1,246 centromeres.
1,246.
Yeah, that's about half of all possible centromeres in these 65 individuals. An incredible leap forward for these really difficult regions.
It's amazing coverage. What about the variation? Was it as crazy as people suspected?
Even crazier, maybe. The most striking finding is the tremendous variation in the length of these centromeric repeat arrays, up to a 37-fold difference for chromosome 10.
37 times difference in length for the same chromosome centromere.
Exactly, just massive structural differences. They also identified over 4,000 new alpha satellite HOR variants, basically new arrangements of the building blocks, and totally novel ways these arrays were organized.
Like what?
For instance, on chromosome 1, they found an insertion that effectively split the centromere into 2 distinct alpha satellite arrays.
Split the centromere?
Functionally split the main repeat array, yeah. Revealing unforeseen complexity in how chromosomes are built.
And then there's this dikinetochore thing. This sounds pretty wild. Can you explain that?
It really is wild. So the kinetochore is the protein machine that builds onto the centromere. It's where spindle fibers grab on during cell division to pull chromosomes apart. Super important.
Right.
They looked at chemical marks, methylation patterns along each centromere, and they found that 7% of the human chromosomes they analyzed showed evidence of 2 distinct regions with the signature of being kinetochore ready.
2 potential attachment points on one chromosome.
Suggests the presence of dekinetotropes, yeah. 2 potential sites for kinetochore attachment.
Whoa! What are the implications of that for chromosome stability, health?
That's the huge question. It's a fundamental discovery about chromosome structure. It could really impact how we understand cell division, maybe genetic disorders where chromosomes get missegregated. We just didn't know this could happen commonly.
And were mobile elements found in these centromeres too? Could they be involved in this?
Oh, yes, good point. About 30% of these centromeric repeat arrays contained at least one mobile element insertion, mostly L1s and Alice.
Where were they located?
Most were near the edges, the periphery, but some were found right between those potential kinetochore sites in the dekinetochore cases.
So maybe influencing the structure?
Possibly. Maybe they're involved in breaking a single kinetochore into 2, or maybe they act as boundaries. It definitely hints at a potential role for them in centromere function and evolution, which is a whole new area to explore. V, the innovation unpacked. What this means for biology.
Okay, stepping back. Why was this ambitious project to map the dark regions so important in the first place? What big problem was it trying to solve?
Well, the core motivation really was to tackle the missing heritability problem we see in genome-wide association studies, GWAS.
Right, where genetic studies find links to disease. But we can't explain all the genetic influence.
Exactly. For years, GWS signals kept pointing to regions of the genome that were poorly mapped, full of gaps.
So researchers couldn't follow up properly.
Right. Clinicians, researchers, they lacked the proper reference sequences to really interpret those genetic links to disease. The hypothesis was simply that missing sequence itself accounted for a big chunk of this dark variation heritability.
And did this study support that?
It largely confirms that hypothesis, yeah. By filling in the map, we can now see many of those missing pieces and understand their links to disease much better.
Now, this wasn't just about throwing more data at it, right?
Yeah.
You mentioned innovative methods. Can you highlight some key technical innovations that make this possible?
Absolutely. It was very innovative. One key thing was adaptive sampling on the sequencer, the Prometheon. This let them specifically target and enrich for repetitive regions without needing PCR amplification first.
Avoiding bias from copying the DNA?
Exactly. PCR bias often messes up analysis of repeats. Another huge leap was graph-based assembly using tools like Virgo.
What does that do?
It can stitch together incredibly complex regions, like centromeres, where the 2 parental copies are different, by using Hi-C data, which tells you which bits of DNA are close together in 3D space.
Well, door.
Yeah. And they use trio-aware hypheism for figuring out which variant came from which parent using data from parents when available. This helped ensure they could deliver those chromosome-spanning maternal and paternal haplotypes. Really sophisticated computational stuff.
And what about the big picture for our genetic references? How do these innovations change the fundamental tools genomics uses?
Well, their pangenome graph construction is huge. They aligned all these new assemblies, and it produced 14 million new nodes in the graph.
Nodes representing what?
Representing insertions, chunks of DNA, completely absent from the old GRC-H38 reference.
14 million new pieces of human DNA sequence found.
Basically, yeah. It also flagged 13,350 genes where they found previously unannotated exons, the protein coding bits, meaning we're still discovering the basic structure of our own genes. And then there's the ClinVar uplift. They took known pathogenic variants from the ClinVar database and relocated over 2,000 of them onto the new correct coordinates in the P22 reference.
Fixing mistakes from the old map.
Fixing mistakes, correcting misassigned risk alleles, even revealing 9 ghost recombination hotspots that were invisible before. These technical improvements have direct, tangible clinical implications. Genetic diagnostics just got more accurate.
This really does sound like a true paradigm shift.
Yeah.
Moving away from a single flawed reference.
It absolutely is. This effort genuinely moves us beyond single reference genomics toward a true pangenome, a comprehensive map reflecting all human genetic diversity, not just one average sequence that doesn't fully represent anyone. By finally illuminating these hypervariable regions, subelomeres, acrocentric arms, centromeres, this work fundamentally recalibrates how we interpret genetic variants.
It paves the way for
population-specific reference panels, which are absolutely vital for health equity around the world, ensuring that genomic medicine works for everyone, not just certain populations.
So looking ahead, what does this all mean for the future of biology and medicine?
Well, this incredible resource, this pangenome, it's likely going to underpin future risk prediction models for diseases, improve how we infer ancestry, even help develop more precise maps to check for off-target effects in CRISPR gene editing.
So foundational for many future applications.
Foundational, exactly. Now, challenges remain, of course, like scaling this phasing and assembly to even more diverse, complex, admixed genomes. But the foundation laid by this deep dive into the human genome, it's profound. It's a massive stepping stone to a future where our understanding of human genetic variation is truly complete and equitable.
Wow. From mapping hidden variants and repeats and duplications to unveiling entirely new chromosomal structures like decanonatures, this deep dive has really shown us that our understanding of the human genome is still very much evolving.
Absolutely.
We're moving beyond a single, incomplete reference map to a truly diverse, detailed pangenome that captures, hopefully, the full spectrum of human genetic variation. So what does this new, more complete map of human genetic variation mean for you? Well, perhaps it means that the next time you or someone you know gets a genetic test, the answers might be more precise, more comprehensive, maybe more tailored to the unique variations hidden in your own genome. And it raises this really exciting question. What other fundamental truths about human biology might still be hidden in plain sight, just waiting for even better tools like these ones to finally reveal them?